Good morning, students. Today I'll be taking an introductory lecture on cell signaling. Now, each cell in a multicellular animal is programmed during development to respond to a specific set of signals that act in various combinations to regulate the behavior of the cell and to determine whether the cell lives or dies and whether it proliferates or stay quiescent. Now, all these signals, the entire process, we call it as cell signaling. That is, in this process, a cell responds to the signal. It responds to the signals and alters its behavior according to the signal. So now this cell signaling requires two things. There are two major components, that is extracellular signaling molecules and a complementary set of receptor proteins in each cell that enables it to bind and respond to them in a programmed and coordinated way. Now, if we are talking about signaling receptors, we have two kinds of signaling receptors. One is cell surface receptors, the other one is intracellular receptors. Now, what are cell surface receptors? Now, most extracellular signaling molecules, they are hydrophilic and are able to activate receptor proteins on the surface of the target cells. Now, these receptors which are present on the cell surface, we call it as cell surface receptors. Now, these receptors act as signal transducers, converting the extracellular binding event into the intracellular signals, thus alter the behavior of the cell. Now, the next one is intracellular receptor. As the name suggests, these intracellular receptors, they are not present on the cell surface receptor. So generally, the signaling molecules that are involved with the intracellular receptors, these are small hydrophobic signaling molecules, including the steroid hormones, thyroid hormones, and the retinoids. These diffuse across the plasma membrane of the target cell and activate intracellular receptor proteins, which directly regulate the transcription of specific genes. Now, some dissolved gases such as nitric oxide and carbon monoxide also acts as local mediators by diffusing across the plasma membrane of the target cell and activating an intracellular enzyme. Now we'll see the different types of cell surface receptors. Now there are three main families of the cell surface receptors, which each of which transduces extracellular signal in a different way. So we have three kinds of cell surface receptors. Now the first one is ion channel linked receptor. The other one is G protein linked receptors. And the third one is enzyme linked receptors. Now, what are ion channel linked receptors? These are transmitter gated ion channels that open or close in response to the binding of a neurotransmitter. So what it does, it leads to the opening and closing of ion channel, and which is generally in response to the neurotransmitters. Now the other one is G protein linked receptors. Here in this one, this G proteins are there, which indirectly are involved in the signal transduction process. And now the third one is enzyme linked receptors, which either act directly as enzymes or are associated with enzymes. Now the first one, as I said, this ion channel linked receptors, these are also known as transmitter gated ion channel and are involved in rapid synaptic signaling between electrically excitable cells. Now, this type of signaling is mediated by a small number of neurotransmitters that transiently open or close the ion channel formed by the protein to which they bind. Now, this leads to the change in the ion permeability of the plasma membrane and thereby the excitability of the postsynaptic cells. Now, the ion channel linked receptor belong to a family of homologous multipass transmembrane proteins. Now, the other one is G protein linked receptors. Now, they act indirectly to regulate the activity of a separate plasma membrane bound target protein, which can be an enzyme or it can be an ion channel. Now, the interaction between the receptor and the target protein, as I said, is mediated by a third protein called a trimeric GTP binding regulatory protein. In short, we call it as G protein. Now, the activation of the target protein either alters the concentration of one or more intracellular mediators or alters the ion permeability of the plasma membrane. Now, the intracellular mediators act in turn to alter the behavior of yet other proteins in the cell. All of the G protein linked receptors belong to a large superfamily of homologous 7-pass transmembrane proteins. Now, just G protein linked receptors mediate the cellular responses to an enormous diversity of signaling molecules, including hormones, neurotransmitters, local mediators, etc. So now what happens when an extracellular ligand binds to a G protein linked receptor? The receptor changes its conformation and switches from inactive form to the active form. Now, most G protein linked receptors activate a chain of events that alter the concentration of one or more small intracellular signaling molecules. Now, these small molecules are often referred to as intracellular mediators or intracellular messengers. Now, this in turn passes the signal on by altering the behavior of the selected cellular protein. Now, two of the most widely used intracellular mediators are cyclic AMP and calcium. The third one is enzyme-linked receptors. Now, these receptors, when activated, they either function directly as enzymes or are associated with the enzymes. Mostly, they are single-pass transmembrane protein with their ligand binding site outside the cell and their catalytic site inside. Now, compared with the other two classes, enzyme-linked receptors, they are heterogeneous, although the great majority are protein kinases or are associated with protein kinases that phosphorylate specific set of proteins in the target cell. Like G protein linked receptors, this enzyme linked receptors, they are also transmembrane proteins with their binding domain on the outer surface of the plasma membrane. Now, there are 5 known classes of enzyme linked receptors. These are receptor guanylyl cyclases, which catalyzes the production of cyclic GMP in the cytosol. We have receptor tyrosine kinases, which are responsible for phosphorylating specific tyrosine residues on a small set of intracellular signaling proteins. Then we have tyrosine kinase associated receptors, which are associated with proteins that have tyrosine kinase activity. Then we have receptor tyrosine phosphatase, which remove phosphate group from the tyrosine residue of specific intracellular signaling molecules, and receptor serine threonine kinases, which phosphorylate specific serine or threonine residues on some intracellular proteins. So that's all about the cell surface receptor and its types. Thank you.